Everyone, my name is Yo-Yo. I'm from the City University of New York in computer science. I'm a second year PhD. I'm very grateful for all of you who choose to stay, although we might be hungry right now for the lunchtime. So I hope you enjoy my presentation and feel free to ask me questions when I'm done. So this work I like to share has a long name called a context-aware deconfounding autoencoder for robust prediction of personalized clinical drug response from cell-like compound screening. I'd like to start with the motivation of this work, like why do we want to do this in the first place? As we know, drug screening is limited to disease models, usually like cell lines. However, those drug response on the disease models does not actually reflect that's a human. So you see there's a natural knowledge gap between this like in vitro to in vivo. So how do we fill up this gap? So we want to utilize the power of deep learning models to accurately predicting the patient-specific response to a new compound, and we think it's critical to the personalized drug discovery. However, there are challenges that you see the patient data often very scarce, and it's hard to train a well generalized machine learning model. And although there are existing models, there might be like they're not reliable enough due to some data heterogeneity or like distribution shift. Also, the omics data often contain a lot of bias and confounding factors like batch effect or different genders, etc. So how do we do this? We have developed this novel context-aware deconfounding autoencoder that can extract the intrinsic biology signals and while removing those context-specific patterns and confounding factors. So here's the rationale of CODE. So you see this picture, the kind of abstract, because we see like there's a source domain drug biomarker and target domain drug marker. They can be the source domain can be our cell line data, and the target domain can be our patient data. And the drug biomarkers are usually the drug target or their gene regulatory network. And our model is to extract the common drug biomarkers, which will be the shared gene regulatory network, and while detecting those unique features for each one of the domains. And those unique features might be contributed to our downstream tasks, like in this case, like the drug response prediction. So here's an illustration of our model. So given label data, which the label means like drug response is known from the cell line. So our goal is to predict those unknown drug response from the patient samples. You see there's a question mark there. So how do we tangle this? Our model has conceptually three stages, and I'll explain them one by one. The first stage, is unsupervised pre-training, and you see the unsupervised is mean to be no labels at all. So where our model takes the unlabeled data from both domains, from the cell lines as well as the patient, and where the model maps them into this embedding space in the middle, and where the biological confounding factors are disentangled as and also you see the embeddings from the patient is also well aligned with the embedding from the cell line sample, like so. And then once you get this nicely well aligned embeddings, it's further passed to the next stage, which is supervised training. So we also call this fine tuning because you see like we finally use the label data, which is the known drug response data, to tune the model trained from the stage 1, where all the the weights are getting updated by the label data. And then comes to the last stage, which is the inference stage, where given the patient sample, you firstly using this pre-trained model to get the confounded and well aligned embedding of this patient, and then we use this fine-tuned model to do the final prediction of this drug response of this given patient. I'd like to have this pointing the laser. Yeah, got it. Okay, so I can point. So you see this architecture. So this is a base architecture of our model. So in the middle, there's a shared encoder which extract the common intrinsic signals from both domains, and each domain has their private encoders, so to learn their private embeddings locally. And the decoder here is shared for both domains, where this decoder takes the concatenation of the private and the shared encoders of each domain, and then do the try to reconstruct the input, like the the whole concept of autoencoder, and by this reconstruction loss here. So this reconstruction loss, although there's only one, but actually there's like we concatenated the two construction loss together. So there is one MSE, there is another MSE. So yeah, and then you see this. We also have this local loss function between the private feature and the shared private encoder and a shared encoder. So try to push apart this private and shared. So it's like a squeeze and push kind of game to get like more common features while detecting the unique features. So this function, this loss function will use the orthogonality loss here for both domains. And then these learned shared encoders are further appended with another classification network, which will be trained during this fine-tuning stage, as well as using in the final prediction. Our models also has two variations. They're majorly varied on the way to concatenate of the private and shared encoders. So we use two different. Instead of simply concate them together, as I just showed in the base architecture, we use different loss function to just try to make those two embeddings as similar as possible. So first one we use MMD loss, and another one we use adversary loss, which is also commonly seen from those computer vision task on deep learning. So this loss is in the format of min-max optimization between this is a critic network and also with the encoder components, as you see. So here's the result, which is the exciting part, and this is a very straightforward result. So you see this TSNE plot. The left one is the original plot. The next one is the plot which use the embeddings from which is the shared encoder to generate the new samples on the both domains from TCGA, which is a patient, and the CCLE, which is cell line. So you see the original, they're like randomly distributed. However, the one generated by our shared encoder, which contains the shared features of them, and they're well aligned and very overlap together. So which also shows our model does a good job in aligning both domains together. So here's another result. We compared our model with the baseline models using this ex vivo drug response from this PDTC dataset. And you see our model among all the baseline models, it's the best performance on the average ranking of both AURC and the AUPRC, which is the common using binary classification metrics. And another one is another test we perform is to predict the chemotherapy. And again, our model consistently outperform the the baseline models on all the metrics. So here's the application of our model on the personalized medicine. We apply the spectral biocluster to divide the drugs into 30 clusters from the predicted drug response matrix. And you see the darker, the stronger the response is as the heat map. Among this, all the drugs tested, top 3 most responding drugs are here. As you see, the darkest color, they're they're also the top 3 predictive drugs based on the clinical study. So this Gefinet is EGFR tyrosine inhibitor, and this ASER can block the growth of cancer cell harboring the activated EGFR mutate. However, this gemcitabine is a treatment which is for the cells are not harboring the EGFR mutation. So consistent with the clinical study, you see this prediction here for these 3 drugs. The ones from the first 2, like Gefinet and ASER, they're similar, however different from this gemcitabine. So which is consistent, which is good. So to summarize this paper, we introduced this transfer learning framework called AE to predict individual patient drug response from the model trained from cell line data. And also performed benchmark studies, which demonstrate our model is over state of art, and the prediction of drug response and the TGC are largely consistent with the existing clinical observations. And there are a couple of directions for the future work. For example, the sophisticated methods can be used to integrate the data, and the integrity of our model needs to be improved. And we think that estimating of the uncertainty for each new case can further improve the performance of the model, and may be critical for the clinical applications. And sorry, I run out of time, but I like to thank my dearest professor who's sitting there, and also my other coworkers in this conference, and our founders. Thank you for staying at the last minute. Thank you so much.